Hello, my name is Dena Leshkowitz. I'm head of the Bioinformatics unit at the Life Sciences Core Facility in the Weizmann Institute, and I will talk about UTAP2, user-friendly transcriptome and epigenome analysis pipeline, mostly developed by Dena Leshkowitz, which is our programmer. So UTAP is an open source, web-based, intuitive platform currently installed in the Weizmann Institute cluster and used extensively by its researchers. In addition, it is available as an open source application for the biomedical research community in order to enable researchers to efficiently and accurately analyze transcriptome and epigenome sequence data. UTAP supports the transcriptome pipelines, starting from cDNA sequences and ending with gene counts and differentially expressed genes. The outputs are placed in a structured folder, and the results are summarized in a rich and comprehensive report. We also support epigenome pipelines, ChIP-seq and ATAC-seq. UTAP installation is a singularity image which contains Miniconda environment with all the software and applications required for running the pipelines and the ability to leverage from parallel cluster resources. So the pipeline starts from for RNA-seq for transcriptome analysis, starts by the cDNA sequences, trims the sequences, maps them to the genome, makes the count matrix, and computes the differentially expressed genes, ending with a comprehensive report containing also quality control information. We also support protocols in which UMI sequences are inserted. We also support epigenome pipelines for ChIP-seq and ATAC-seq, starting from the genomic fragments and mapping them to the genome, calling peaks, and again, ending with the reports with also quality control information. So as we promised, the UTAP application is a user-friendly application, meaning it has a web graphical interface, which allows an easy setup of the pipeline. Here I'm showing several screenshots of the interface, starting by a screenshot that shows the initial step of defining the project, where the sequences are located, to which genome we should map the sequences, and your email in order to notify you that the pipeline has finished. The samples are put in folders according to the sample name. So once you told us where the folders are, basically we already know the sample name, and then you can drag the samples into the category names or category groups that you have labeled here. So basically by clicking here, LPS or control in this case are our categories, and we drag the samples into the relevant category. We also can perform batch correction. The user needs to notify which sample is in which batch by just clicking the certain batch and the sample name. There are also additional parameters, advanced parameters that the user can change, and then of course run the analysis. So the reports are rich, contain a lot of information, starting from sequencing and mapping quality information. So here we see the reads sequence quality, and then the number of reads that we have per sample as we step through the pipeline. Here we can see the coverage, the reads coverage on genomic regions. The genomic region is the gene body. Some exploratory analysis in order to show us which are the top expressed genes, heat map showing us which samples are more correlated to each other, and again, a dendrogram showing which samples are more similar to each other. We also have plots in order to see information regarding the differentially expressed genes. So there is a table which allows you interactively to view a certain gene dot plot, expression dot plot, or its position in an MA plot. Here we can see a volcano plot. In green are the differentially expressed genes. This is also an interactive plot. And we produce a heat map of the differentially expressed gene expression level. So UTAP is available to be installed on various platforms, on your local Linux server, on a cluster that uses LSF, and we're working on having it work also on the cloud. Please read the docs information about how to download it. Our previous version of UTAP was published in 2019 and has many citations from people that have used it, mostly from the Weizmann Institute. So thanks for listening, and I'm available to answer questions and also give you a live demonstration. So please contact me, and I hope you found this enjoyable, this explanation, and hope to see you using UTAP in the future. So goodbye.